Hello, I am Shafir Rahman, and today we are going to discuss about acute leukemias. Now, before going into the detail of what acute leukemias are all about, first of all, let us define the term leukemia. Now, leukemia is basically the malignant proliferation of white blood cells. So, it's a malignancy of white blood cells, and mostly the cells when appear in the blood we are calling them leukemia. So white blood cells when appear in the blood we are calling that leukemia, in contrast with lymphoma, in which the malignancy of white blood cells is present in the lymph nodes. So leukemia generally is defined as malignant proliferation of white blood cells present in the blood, and in contrast with lymphoma, in which that's the malignancy of white blood cells inside the lymph nodes. So the hallmark feature is the white blood cells, and it's a white blood cell disorders, and white blood cells are is an umbrella term in which we have multiple cells, which we will discuss one by one. Now, you can see that classification of leukemia can be done in numerous ways, and one of the way is by differentiating between myeloid and lymphoid lineages, which we'll discuss shortly. Then acute versus chronic leukemias. So the differentiating point between acute and chronic leukemias is the characteristics of cells which are found in the peripheral smear. So for example, in acute leukemias, by term acute we means rapid onset of the symptoms, but in acute leukemia we are seeing blasts in the bone marrow. So the term blast is basically, it's a precursor to mature circulating blood cells. So blasts are precursors to the mature circulating blood cells such as neutrophils, monocytes, lymphocytes, or erythrocytes. So they are usually immature cells which are precursor cells to the much more mature cells. So when we are finding them in the peripheral smear, so we are saying that it's an acute leukemia. But when you contrast it with chronic leukemias, so we are not finding the blasts, but we are finding some mature cells, much more mature cells than the blasts. And the onset of the symptoms will be much slower than the acute leukemia. So the differentiating point between acute and chronic leukemia is the presence of blasts in the acute leukemia as opposed to chronic leukemia, in which we are seeing much more mature cells. Now if we can see the hematopoiesis, so first of all, inside the bone marrow, we are having that multipotential hematopoietic stem cell, which is also called hemocytoblast. So from that stem cell, it can go along the two lineages. There are two different fates of that multipotential hematopoietic stem cell, and one of the fate is to become common myeloid progenitor, and the other fate is to become common lymphoid progenitor cell. So if it becomes common myeloid progenitor cells, so common myeloid progenitor cell then can be converted into megakaryocytes, and hence further differentiation into the thrombocytes. It can convert, common myeloid progenitor cell can convert into erythrocytes, can convert into mast cells, and can convert into the myeloblast. And myeloblast comes under, like, it is produced by common myeloid progenitor cells. So what myeloblast will do, that it can differentiate into the basophil lineages, neutrophil lineages, eosinophil lineages, or it eventually convert into the monocyte macrophages lineages. So that myeloblast seems to be a very important cell, and we will discuss in acute myeloid leukemia and chronic myeloid leukemia that what sort of cells can eventually grow out in those leukemias. So similarly, the other fate which we have talked about is that it can convert into the common myeloid progenitor cell, like multipotential hematopoietic stem cell can convert into the common lymphoid progenitor cell, and that common lymphoid progenitor cell can become natural killer cell, which is a large granular lymphocyte, or it can convert into the lymphocyte lineages, in which eventually can become B cells and T cells, and T cells can become into CD4 positive T cells or CD8 positive T cells, and B cells can convert into the plasma cells and antibody secreting cells. So the whole process of hematopoiesis is going on in the bone marrow, and eventually that's the place from which leukemias are emerging out. So if we can see that myeloid disorders, which are uh, uh, which comes out of that common myeloid progenitor cells. So if it's inside the bone marrow and myeloblasts are proliferated abnormally, we will call that acute myeloid leukemia. Then, similarly, if red cells are overproduced, like we have said previously, that common myeloid progenitor cell can convert into erythrocytes, which are the red blood cells. So if that cell is abnormally proliferative, so we call it the famous polycythemia vera. If the platelets are overproducing, the megakaryocytic lineage is overstimulated and malignancy is occurring, we will call that essential thrombocytosis. Then the granulocytes can become abnormally proliferative and can produce that chronic myeloid leukemia, which we'll discuss later on. So myeloblasts can become proliferative or malignant, can become malignant inside the bone marrow. They can be called acute myeloid leukemia, or inside the blood red blood cells can proliferate and can cause polycythemia vera. Granulocytes can become abnormal and we will call them chronic myeloid leukemia, or platelets can overproduce and we will call that essential thrombocytosis. And these three disorders, polycythemia vera, chronic myeloid leukemia, and essential thrombocytosis, come under the umbrella of myeloproliferative disorders, which we will discuss in the next podcast. So if we can see what are the granulocytes, granulocytes are a type of immune cells that has granules with enzymes that are released during infections, allergic reaction, and asthma. For example, neutrophils, eosinophils, and basophils are granulocytes. So when those cells are overproducing, so will say that it is a chronic myeloid leukemia. And when the myeloblasts are proliferating inside the bone marrow, we will call that acute myeloid leukemia. Now coming towards the lymphoid disorders, if we can see that it's a bit complex, that from the bone marrow lymphoblasts can be produced, then they go into the bloodstream and can convert into T cells or B cells depending upon the marker which is present on their membrane, or they can be produced inside the lymph nodes during the maturation process. So when in the bone marrow lymphoblasts are overproduced or malignancy of the lymphoblast in the bone marrow, we will call that acute lymphoblastic leukemia. So those cells are basically immature cells, which what we have called about earlier, that the blasts are immature. So whenever the term blast will come, we will say that it's the immature cells produced from bone marrow. So when inside the bone marrow there is a malignancy of the lymphoblast, we will call that acute lymphoblastic leukemia, or the classical ALL. Then in the bloodstream, if it becomes a little bit mature and there is a malignancy of a bit mature B or T cells, we will call that chronic lymphocytic leukemia. And then the lymphoma is basically the malignancy of T cells or B cells inside the lymph nodes, and we will discuss Hodgkin and non-Hodgkin lymphoma in the later podcasts. And then B cell can be converted into the plasma cell antibody secreting cells, and that's the classical myeloma and plasma cell disorders, which we will also discuss later on. Now we will start discussing the acute lymphoblastic leukemia, which is classically called ALL. So previously discussed that blasts are immature cells found in the bone marrow and then can go into the bloodstream, and then the presence of blasts will let us know that whether it is an acute leukemia or chronic leukemia. So acute lymphoblastic leukemia is basically the presence of lymphoblasts in the bone marrow and blasts are found. So the first thing first, that it's the epidemiology of the disease is that it's mostly found in children. ALL is mostly found in the children, and the peak incidence is at the 4 years of age. Usually presents with the fever, bone pain. Now, bone pain is occurring because when the blasts are proliferating inside the bone marrow, so it will call marrow expansion. And when the marrow will expand, so eventually the pain signals will stimulate the pain receptors, and hence the bone pain can occur. Then when the blast cells grow to a sizable margin, when it can infiltrate the other organs and come into the bloodstream and can start infiltrating the other organs, so ALL can present as lymphadenopathy, splenomegaly, and hepatomegaly. So when lymphadenopathy, splenomegaly, or hepatomegaly is occurring, it means that there is infiltration of these blasts or lymphoblasts um, um, in lymph nodes, spleen, and liver. Similarly, these lymphoblasts can spread to the meninges, and when it will spread to the meninges, the classical symptoms of headache and vomiting can occur. And eventually, it will overcrowd the bone marrow, and it can disrupt the production of other cells, like, for example, it can disrupt the production of red blood cells and can cause anemia. It can disrupt the production of platelets and can cause thrombocytopenia, or it can disrupt the production of granulocytes and can cause neutropenia. So the abnormality of one cell initially will uh, start making other cells abnormal as well. So initially it was basically the disorder of the lymphoid lineages, but now it's also disturbing the red blood cell production, granulocyte production, platelet production. So we can have diverse range of symptoms along with fever, bone pain, lymphadenopathy, or splenomegaly or hepatomegaly. We can have that bleeding disorder, we can have that weakness and fatigue due to anemia, and we can have infections due to neutropenia. So when we see peripheral blood or peripheral smear with acute lymphoblastic leukemia, so the hallmark feature is the presence of lymphoblasts. So the lymphoblast can appear like a myeloblast because the immature cells, you can't differentiate immature cells just on the basis of the microscope, because most of them have the same morphology. But we have some special tests that we can distinguish lymphoblasts from the myeloblasts. So if we can recall our immunology course, that when the lymphocyte is developing in the development of when lymphocyte is occurring from a stem cell, so the most important enzyme is which is present in the immature lymphocytes is known as terminal deoxytransferase enzyme, which is also known as TDT plus. So TDT plus is present in the immature lymphocytes, and it is the hallmark feature of lymphoblasts. And through the presence of TD plus, we can differentiate it versus myeloblast, in which there is absence of TDT plus, and the markers of myeloblasts are different, which we will discuss shortly. So the stem cells can convert into either B lymphocytes or T lymphocytes, but during its immaturity we will have the TDT positive enzyme, and which is basically a DNA polymerase, and it will help in the maturation process of the lymphocytes. So if we recall that the T cells lymphocytes usually have that CD marker ranging from 1 to 8. It's that single number digit of cluster of differentiation which is present on the T cells. So T lymphocytes usually have CD markers from 1, 2, 3, 4, 5, 6, and 7, 8. However, the B cells are usually associated with double digit CD markers like CD10, CD19, CD20, 21, 22, 23. So in that way the lymphocytes get matured, that either it will become double digit CD like double digit CD marker like 19, 20, 21 and will be called B lymphocyte, or it can convert into a single marker CD like CD1, 2, 3, 4 and can become like T cell, mature T cell. So acute lymphoblastic leukemia is usually pre-B cell malignancy. Now Pre B cell malignancy means that B cell is not matured